So this is our fourth video under the topic cell signaling, and today we are going to talk about intracellular receptors. Earlier we had covered about extracellular receptors and signals or ligands, and the link will be provided in the description below. So what are intracellular receptors? As the name suggests, these are receptors present inside the cell, and they bind to ligands or signals which enter into the cell. The receptor binds to the ligand and they form the receptor-ligand complex, which translocate into the nucleus and then help in the transcription of the specific DNA segment and later translates it into proteins. And this is the specific response for the ligand. So now let's see the various ligands which bind to intracellular receptors. The first type is the steroid hormones, like sex steroids or corticosteroids. So what are steroid hormones? They have the steroid core, that's the 17 carbons bonded in 4 fused rings. So steroid hormones enter into the cell by simple diffusion. In mammals, there are 6 families of steroid hormones, like estrogen, progesterone, androgen, mineralocorticoids, glucocorticoids, vitamin D. So what are estrogens? These help in the development and regulation of the female reproductive system and secondary sexual characters. Progesterone is responsible for the menstrual cycle, pregnancy, and embryogenesis. Androgen helps in development and maintenance of male characteristics. Mineralocorticoids influence the water and electrolyte balance in the body. Glucocorticoids' functions include reducing inflammation and immunosuppression. Vitamin D is responsible for increasing intestinal absorption of calcium, magnesium, phosphate, and many other biological effects. So the first 3 are commonly called sex steroids, and the other 2 are called corticosteroids. So these are mainly produced by gonads and adrenal gland. So we have seen the first type of ligand, that is steroid hormones. The other types of ligands which bind to intracellular receptors are thyroid hormone T3 and T4, lipophilic vitamins or fat-soluble vitamins like vitamin A and vitamin D, small molecules like nitric oxide, hydrogen peroxide, etc., and secondary messengers like cyclic AMP, cyclic GMP, and calcium, etc. So all these bind to intracellular receptors. Now let's see the type of receptors. Receptors are mainly of the type nuclear receptor. These are basically transcription factors. Another type is the receptors present on the membranes of the intracellular organelles like IP3 receptor, especially endoplasmic reticulum. So what are nuclear receptors? They are able to directly interact with and control the expression of genomic DNA. That's why they are called nuclear receptors. They may be present either in the nucleus or in the cytoplasm, but the effect is directly on the genomic DNA, and that's why they are called nuclear receptors. Nuclear receptors belongs to transcription factor superfamily, so they're basically transcription factors, and they control a wide variety of biological processes including cell proliferation, development, metabolism, reproduction, etc. So nuclear receptors have this common structure. That is, the receptor has an amino terminal and a carboxy terminal. And this amino terminal, it interacts with transcription factors. So they have transcriptional activation functions. And then, since this is a receptor, of course, it will bind to a ligand or a signal, which could be a hormone. So it has a hormone binding site. And the effect of this receptor hormone complex will be directly on the DNA. So of course it has a DNA binding site. So this domain is called A or B domain, and this is called the C domain. And then you have D, E, F domains, which are not mentioned here. So first let's see the AB domain. This is called the amino terminal or AB domain. So it's highly variable in both sequence and size, and in some cases it harbors potent transcriptional activation functions. So it interacts with transcription factors. So The next domain is the C domain or DNA binding domain, and of course it binds to the specific sequence of the DNA. And then there is a hinge domain or the D domain. There's a D domain or the hinge domain present here, which targets the protein to the nucleus. That is, it has the nucleus localization signal, NLS. And then you have this carboxy domain or the ligand binding domain or hormone binding domain. So this is the E domain, and sometimes there is an F domain, that's a highly variable carboxy terminal tail. So this is the common structure for all nuclear receptors. Now let's see the different types of nuclear receptors. There are mainly of two types, type 1 and type 2. Type 1 receptors are mainly present in the cytoplasm, and they bind to ligands like steroid hormones, androgens, progesterone, estrogens, glucocorticoids, mineralocorticoids, etc. And the type 2 receptors are present in the nucleus. So they are already in the nucleus, but in an inactive state, may be bound to co-repressor molecules. So the ligands that bind to type 2 type of receptors are thyroid hormones, vitamin A, vitamin D, and small molecules like nitric oxide. So let's see in detail. Type 1 receptors are present in the cytoplasm. They include steroid hormone receptors like androgen receptor. So the receptors are usually held in an inactive conformation by inhibitory proteins like chaperones, heat shock proteins, etc. So when the ligand binds to the receptor, there's a conformational change, and then this causes the inhibitory protein to dissociate from the receptor. Now the ligand receptor complex translocates into the nucleus. So the receptor binds to the DNA with its DNA binding domain, and it binds to the promoter region of the target genes. In the case of hormones, it's called hormone response elements or HRE. Now this can be facilitated by the association of other proteins like coactivator proteins. Let's see some examples. Coactivators are transcriptional cofactors that help to increase the level of gene induction. Example, histone acetylase, which facilitates the transcription by increasing the ease of unraveling of DNA from histone proteins. Then you also have co-repressors that bind to the receptor and repress gene activation, like histone deacetylase, which prevent the further transcription by tightening histone interaction with the DNA. So now we have talked about type 1 receptors present in the cytoplasm and which binds to steroid hormones like sex steroids and corticosteroids. Now let's move on to type 2 receptors which are already present in the nucleus. The first type is the thyroid hormone receptor. So these are bound to DNA response elements within the nucleus, but the receptor acts as an active repressor of transcription. Only when the thyroid hormone or the ligand binds to the receptor, it activates transcription of the specific genes. Now the other type is peroxisome proliferator activated receptor or PPAR family. These are also found in the nucleus, but they are bound to co-repressor proteins. Only when the ligand binds, the receptor is liberated. So what are PPAR family? These receptors function as sensors for endogenous fatty acids like eicosanoids, and they regulate the expression of the genes that influence metabolic events. Now the other type is a retinoic acid receptor. These are also located in the nucleus, and they form complexes with steroid hormone receptors in order to regulate the production of essential gene products. So retinoic acid receptors, they also bind to co-repressors in the absence of the ligand. So retinoic acid receptor binds to retinoic acid, which is formed from the metabolism of vitamin A. Now let's see vitamin D receptor, VDR. They bind to their ligand calcitriol, which is the active form of vitamin D, and they form a heterodimer with RXR, that is retinoic acid related receptors. That is, most of the receptors, they form a homodimer or heterodimer and then cause the transcription. So here VDR binds to the VDR responsive elements in the genomic DNA. This results in the expression or transrepression of specific gene products. So I said receptors can exist as monomers, homodimers or heterodimers. Steroid receptors usually form homodimers, like estrogen receptor binds to another estrogen receptor and then they continue their action. But most of the type 2 receptors form heterodimers, usually with RXR, that is retinoic acid related receptors. So now let's see what are the full form for these abbreviations. AR is androgen receptor, ER is estrogen receptor, and GR is glucocorticoid receptor, MR is mineralocorticoid receptor, and RXR is retinoic acid related receptor, RAR is retinoic acid receptor, etc. So now I think we should talk about ligand-gated ion channels, because these are fundamental signaling molecules in neurons. As we all know, ligand-gated ion channels can be gated by both intracellular and extracellular ligands. So we have to discuss about ligand-gated ion channels in intracellular signaling too. So these molecules are localized on intracellular organelles also. Let's see some examples or some types. Cyclic nucleotide gated ion channels. So the name may look a little difficult, but from the name itself we can identify that these receptors bind to, or the ligand for these receptors are cyclic nucleotides like cyclic GMP, cyclic AMP, cyclic GTP, etc. So these are the ligands that bind to these receptors, and then since this is a ligand-gated ion channel, when the ligand binds to the receptor, the ion channel opens up and the ion current or the flow of ions occur. The ions which flow through this channel are sodium ions and potassium ions. Another type of receptor is IP3 receptor. So from the name itself we understand that the ligand that binds to this receptor is IP3, inositol triphosphate, and the ion current is calcium. That is, calcium ions flow is controlled by this channel, this ligand-gated ion channel. And you have intracellular ATP receptors, and the ligand here is ATP. So when ATP binds to these receptors, the flow of ions that is controlled here is that of potassium. And you have ryanodine receptor. Here the ligand and the ion current both are calcium ions. So that's a rare case. In most of the cases, the ligand which binds to the receptor is totally different from the ions that flow through the ion channel. I think we should talk about IP3 receptors. So this is basically a calcium channel that is gated by inositol triphosphate ligand. So when this ligand, inositol triphosphate, binds to the receptor, the flow of calcium ions through this channel is regulated. Calcium ions are released from the intracellular storage sites, especially from endoplasmic reticulum. So IP3 receptors are very important for cell division, cell proliferation, apoptosis, fertilization, development, behavior, learning, and memory. I think with this we shall wind up the topic of intracellular receptors, and I shall come up with a new topic. Until then, enjoy learning.